Hello, my name is Agnete Kirkeby, and I'm an associate professor at Lund University. I'll talk to you today about the developments on cell therapies for Parkinson's disease. First of all, I'd like to just remind you that cell and gene therapies are rapidly advancing towards clinical trials and now constitute 27% of all new phase 1 clinical trials in Parkinson's disease. And we can only expect to see more of this in the coming years. So when talking about cell therapies, how do these work? Well, we do have one class of cell therapies which are administered systemically. These are the so-called mesenchymal stem cells, which have been proposed to work through immune modulatory effects. We currently have 4 ongoing clinical trials with mesenchymal stem cells around the world. I'm not going to touch any further on these in this talk, as there is limited evidence for the efficacy of these cell products in Parkinson's disease. Other classes of cell products are administered intracerebrally, and here, for instance, we have the neural stem cells, which have been proposed to work through trophic support, mediated by the secretion of various growth factors from the cells after transplantation to the brain. With this class of cells, we currently have an ongoing phase 1/2 clinical trial, which is led by the company International Stem Cell Corporation, together with Melbourne Hospital, a trial ongoing in Australia with 12 patients, all of which have already been transplanted. These patients have received neural stem cells transplanted into the striatum as well as the substantia nigra at various different doses. Preclinical studies have shown that this cell product does not give rise to dopamine neurons after transplantation, but instead gives rise to immature neural stem cells. The company had announced that the results from this clinical trial were to be released last year, but we still haven't seen any data coming out from the trial, and we're expecting the data to come out very soon. Another class of cell therapies for Parkinson's disease, which is the most promising class of cell therapies, are the cell replacement therapies, aiming to replace the endogenous dopamine neurons with new dopamine neurons through transplantation. And this class of cell therapies has shown the most promising preclinical data, and as well as a proof of concept in Parkinson's disease patients, which has been mediated through clinical trials involving transplantation of fetal ventral midbrain tissue. These are clinical trials that have taken place during the 80s and 90s at Lund University, Cambridge University, as well as several sites in the US. Now, although the results from these clinical trials have been very variable, some patients had no effects, other patients had very good effects of the transplant. This is likely due to the various variations in cell survival after transplantation. We still have learned important lessons from these clinical trials. So what have we learned? We have learned that the transplanted dopamine neurons can survive for many years, more than 20 years in the Parkinson's disease brain, even after only a short period of immunosuppression. We've also learned that when these grafts survive successfully, the dopamine neuron replacement can give substantial long-term clinical benefit to the patient. So in short, if we do this right, we can really achieve remarkable results in the patients. These early trials have led to a follow-up clinical trial called TransEuro, which is led by Roger Barker, the chair of this session. This is a trial ongoing at Cambridge and Lund University, again involving the transplantation of fetal ventral midbrain tissue to assess the feasibility and long-term efficacy of this approach. The trial was planned to involve 20 patients, but ended up completing transplantation of only 11 due to the severely limited availability of fetal tissue for transplantation. Then the trial had to stop transplantation at that point because it was so difficult to get enough tissue. So right now the trial is ongoing with assessment of the patients, and results are expected to be released in the end of 21 or early 22. And although we don't have any data from this trial yet, what we have learned is that this approach is very difficult if we want to transplant many patients. And if we do want to transplant many patients, we need to abandon the fetal tissue approach and instead find a new source of cells to produce the dopamine neurons. And here we've learned that, in fact, we can use human pluripotent stem cells and through directed differentiation, produce the dopamine neurons in the dish instead. The pluripotent stem cells can be derived either from in vitro fertilized eggs, this is what we call human embryonic stem cells, or through reprogramming of somatic cells, and this is what we call human-induced pluripotent stem cells. I'm going to tell you about what's ongoing in this very new and exciting field of using pluripotent stem cell therapies for clinical trials. So I told you before about these two clinical trials, one with fetal tissue, one with neural stem cells, and now we're seeing the emergence of several new clinical trials involving the use of pluripotent stem cells for dopamine cell replacement. Some of these are ongoing and some of them are still in the preclinical pipeline. Now, a major development in this field was that we've actually seen the first transplantation results coming out. I'll just mention to you briefly that I'll be talking here about autologous transplantation as well as allogeneic transplantation. And what does this mean? When talking about autologous transplantation, this means that the patient receives cells from him or herself. The advantage here is that this does not require any immunosuppression, but the disadvantage is that it requires manufacturing of a new cell product for every single patient. So in the end, this approach can become extremely expensive. Allogeneic transplantation means that the patient receives cells from a donor. And the advantage here is that the cell product can be manufactured at large scale for many patients. And the disadvantage is that it requires immunosuppression. So I just mentioned to you that we're seeing the first results coming out. And the first report that we've seen published with these pluripotent stem cells is a case report of a single Parkinson's disease patient who received a transplant of autologous iPS cells for dopamine cell replacement. This was a 69-year-old male patient with 10 years of Parkinson's disease and who had a poor clinical management of symptoms with conventional dopaminergic medication. The patient then produced a biopsy of fibroblasts, which was used to convert the cells into iPS cells, and again, then to form dopamine progenitors, which were transplanted back into the brain of the patient. The patient didn't receive any immunosuppressive treatment since it was an autologous transplantation. Now, their team has released this case report showing data from the 12 and 24 months follow-up of the patient. And this data shows that there was a small increase in fluorodopa uptake in the posterior putamen at 12 months, or sorry, at 18 to 24 months after surgery. So one side was transplanted half a year after the other hemisphere. And the report also shows that there was a moderate decrease in UPDRS motor score 24 months after the first surgery. So this data, of course, should be interpreted with caution because it was a single case report, but I think we will be seeing further data coming out from this one patient that was transplanted. In parallel, we also have a trial ongoing at Kyoto University in Japan, again involving an iPS cell product, but this time an allogeneic product. So several patients can be transplanted. 7 patients are included in the trial that all receive the same product. So far, 4 of these patients have been transplanted and 3 are waiting. We haven't seen any data yet from this trial, but the team has released preclinical data showing that the cell product can mediate full functional recovery in rats, as well as produce dopamine-rich grafts, both in the rat and in the monkey. So what else is coming up in this field? We actually have a clinical trial which was just a couple of weeks ago approved by the FDA and which is currently enrolling patients. This is a cell product produced by Memorial Sloan Kettering together with Cornell and Blue Rock Therapeutics. It's an allogeneic human embryonic stem cell derived dopamine cell product. And in contrast to the previous products I've told you about, this is a cryopreserved product, which can enable a large-scale production for many patients at the same time at a lower cost. So this is, of course, an important development in the field. The team has released several sets of preclinical data showing that these cells mediate dopamine-rich grafts in the rat model of Parkinson's disease, as well as full functional recovery on amphetamine-induced rotations. The planned clinical trial will involve 10 patients, will be open label, with a 1-year primary endpoint of safety and a 2-year secondary endpoint of efficacy. In a similar approach, we have a cell product produced and developed by our team at Lund University. Again, this is an allogeneic human embryonic stem cell-derived dopamine product. It's a cryopreserved product similar to the New York clinical trial. In this clinical trial, we will be transplanting 8 patients, again, open label. It will be a younger Parkinson's disease population, 50 to 75 years old, with more than 5 years of Parkinson's disease, but still at mild to moderate stage. We will have a 1-year primary endpoint of safety, as well as a 3-year secondary endpoint of efficacy, and this clinical trial is estimated to start early next year. We've shown in preclinical data that our cell product can mediate dopamine-rich grafts after transplantation to the rat, whereas if you transplant a control cell population, you get absolutely no dopamine cells. And the same is seen on functional recovery, where the dopamine cell product mediates full functional recovery on amphetamine-induced rotations. We also see, which we find is very important, is that these cells mediate full striatal innervation of the rat, so you can see human nerve fibers growing out and innervating the entire rat striatum to a degree which we find is relevant for the distances which are also present in the human putamen. So in summary, what I've shown you today is that we have 6 ongoing trials with cell therapies. 2 more are in the pipeline to initiate in 2022. What we're currently seeing is the transition going from fetal tissue towards off-the-shelf stem cell products for use in many Parkinson's disease patients. Thank you so much for listening.